Okay, so disaccharides. So di simply means two, and saccharide is sugar. So disaccharide simply means two sugars, the simplest of the basic meaning. So a disaccharide will contain two monosaccharide rings that are joined by what is called the glycosidic linkage. So this glycosidic linkage and the rings, they will consist of a central carbon or a central oxygen that is covalently bonded to two carbons, one in each ring. So we're going to see how this glycosidic bond is formed. So it consists of a central oxygen that is covalently bonded to two carbons, one carbon in each ring. So these disaccharides are formed by the process of condensation or dehydration reaction. Remember when we looked at macromolecules, we defined and talked more about what a dehydration or a condensation reaction is. So these disaccharides are formed by the condensation reaction that occurs between the two monosaccharides and henceforth are joined by the glycosidic bond. So this is an example that is showing us the formation, how it occurs to form this glycosidic bond. So remember, this is an oxygen atom bonded covalently to two carbons, one on each ring. So we have this ring and we have this ring, and we have that covalent bond and that covalent bond. And on this one, we have this ring, this covalent bond, and that covalent bond. So remember also here, this is alpha. This is an alpha glycosidic bond, alpha glycosidic bond. This is beta glycosidic bond. So looking at the direction, just look at the direction, you know that, okay, this is beta, this is alpha. So alpha glycosidic bond. And then you look at the carbons that are reacting or taking part in the reaction. So if it is carbon 1, as in the formation of maltose from two alpha glucose molecules, two alpha glucose, it's carbon 1 of one ring bonded with carbon 4. So carbon 1 and carbon 4 leads to the formation of this glycosidic bond. So it means this will be called alpha 1, 4 or alpha 1 to 4 glycosidic bond. So alpha 1, 4 glycosidic bond. So this is alpha, and then this is a formation of lactose between galactose, beta galactose, and beta glucose. So you have the OH group on carbon 1 of beta galactose and the OH group of carbon 4 on beta glucose, which are going to be involved in the formation of the glycosidic bond. So this will be beta. So this is going to be beta 1, 4 glycosidic linkage. So beta 1, 4 glycosidic linkage. So this is a dehydration or condensation reaction. Remember, water is given out. Water is given out as a byproduct, and we have lactose there. So beta galactose plus beta glucose will give us the disaccharide lactose. Alpha glucose plus alpha glucose, through the condensation reaction, water is given out, and we have the disaccharide maltose, disaccharide maltose. So we're going to understand more when we look at the individual sugars, and how, as you can see, this carbon 6, well, for this reaction to take place, this glucose is flipped. So when you look at the disaccharides, they mainly serve as transport molecules in plants, and they also provide nutrition in animals. So they are temporal energy stores. So what happens is that when energy is needed, these are broken down by hydrolysis reaction. Remember that the hydrolysis reaction we explained in the macromolecule class. So when these are broken down, they're broken down into their different monomers. Then those monomers, for example, glucose, will then be used to produce energy through the process of respiration. The most common disaccharides that we have are sucrose, lactose, and maltose. So sucrose, lactose, and maltose. So sucrose also is a common table sugar, and it consists of a glucose unity, so a glucose monomer combined with fructose. So glucose plus fructose, will give us sucrose. Then lactose, which is a sugar that is present in milk, will consist of one molecule of glucose and one molecule of lactose. So glucose plus one molecule of glucose and one molecule of galactose. So glucose plus the galactose will give us the lactose. And then the desaturated maltose or malt sugar will consist of two covalently linked alpha glucose units. So alpha glucose monomer 1 plus alpha glucose monomer 2 will give us maltose. The sugar is crystalline, they are sweet to taste, and they readily dissolve in water. So these are the disaccharides. We have said that they are formed through the process of condensation, and they can also be hydrolyzed. So they can be split by the addition of water into two monosaccharides unit. So for example, during digestion, during the process of digestion, we find that maltose is hydrolyzed to form two molecules of glucose. So maltose plus water will give us glucose plus glucose. So the hydrolysis reaction will give us back the monomers that are used to form these disaccharides. And now when you look at sucrose also can be got as cane sugar. It's composed of, as I have already mentioned, of glucose and fructose. And these are linked through 1,2 glycosidic bond. So it's C1 of alpha glucose and carbon 2 of beta fructose. So carbon 1 of alpha glucose and carbon 2 of beta fructose, forming a 1,2 glycosidic bond. So the 1 and the 2 simply implies the carbons that are involved, the positions. So the molecular formula for sucrose is C12H22O11, C12H22O11. So when we look at this sucrose, this is the form that most plants actually use to transport glucose. And it is also the sugar that most humans and other animals actually eat. So we find that things like sugarcane are very rich in sucrose. So things like sugarcane are rich in sucrose. So in plants, it's most abundant in the plants. It's translocated in large quantities through the phylum tissue. So sucrose is a non-reducing sugar. So sucrose is non-reducing sugar. because it is joined through carbon 1 of glucose and carbon 2 of fructose that contain the aldehyde and ketone groups, which have the reducing properties in the individual open chain structures. So when we look at the aldehyde group and the ketone group, so you have the aldehyde group in glucose is on carbon 2, and the ketone group in fructose is on carbon 2. So carbon 1 of glucose, that's where we have the aldehyde group. Carbon 2 of fructose, that's where we have the ketone group. Now we find that this glycosidic bond happens between these two carbons, which means that these groups have been involved in formation of that glycosidic bond, and henceforth it makes sucrose to be a non-reducing sugar. Remember that these groups are needed for a sugar to have the reducing properties. So glucose is non-reducing sugar. Then in the industry of importance application, sucrose is used as a sweetener in baking or confectionery, and also as a food preservative. So those are industrial applications. And now this is the formation of sucrose. So remember I said alpha glucose plus beta fructose. So we have carbon 1 and carbon 2. Carbon 1 and carbon 2. Through the process of condensation, of dehydration, will lead to the formation of 1,2 glycosidic bond, 1,2 glycosidic bond. When this sucrose molecule is broken down through the process of hydrolysis, it's going to give us these monomers, the individual monomers. We're going to have alpha glucose and beta fructose, alpha glucose and beta fructose. So the bond that is formed in sucrose is 1,2 glycosidic bond. And then this is simply the same reaction. And looking at the reaction, remember it's alpha glucose react with beta fructose. So this is going to lead to the alpha 1. So remember it's carbon 1 of glucose, alpha 1, beta 2, remember it's beta fructose and position 2. So alpha 1, beta 2 glycosidic linkage. So this bond in full, it's alpha 1, beta 2 glycosidic linkage, the one we find in sucrose. Then we have lactose. Lactose, also called milk sugar. So this one is composed of galactose, which is linked through one beta 1,4 glycosidic bond. So it simply means that this is a reaction between C1 and C1 of beta galactose and C4 of glucose. So beta 1,4 glycosidic bond. So it has the same formula as sucrose, C12H22 or 11, and it is water soluble as well. Now lactose is a reducing sugar, because when you look at carbon 1 of its glucose, it is free. So carbon 1 of its glucose is free. Remember, the carbon that is reacting in glucose, it's carbon 4. So which means that the carbon 1 of glucose will be free. And henceforth, it can form an open chain to expose the aldehyde, which has a reducing properties. So lactose is a reducing sugar. So this is showing the formation of lactose and breakdown. So this is galactose. This is galactose, and this is glucose. So carbon 1, this carbon 1 of galactose will react with carbon 2. So the reaction takes place between carbon 1 and carbon 4 rather. So carbon 1 of galactose and carbon 4 of glucose through the process of dehydration or condensation. Water is given out, forming the beta 1,4 glycosidic bond. So beta 1,4 glycosidic bond in lactose. So if this lactose is broken down through the process of hydrolysis, I'm going to get the individual monomers of galactose and glucose. So this is lactose. This is lactose. So beta galactopyranosyl 1 to 4. Remember, it's between carbon 1 and 4. Alpha D glucopyranose. Then this is sucrose, the one we talked about. Alpha D glucopyranosyl 1 to 2 or 1,2 beta fructopyranoside. This is simply structures of lactose and sucrose. Now, when you look at maltose, which is also malt sugar, it's water soluble and consists of 2 glucose molecules, which are linked through an alpha 1,4 or alpha 1 to 4 glycosidic link. So this one is it's a reducing sugar. Remember, the carbons that are participating are those of position 4 and 1. So it simply means that the carbon of one of its glucose molecule, the one whose position 4 participate in the formation of the glycosidic bond, is free. So it can form an open chain to expose that aldehyde group, which has reducing properties. So maltose is a reducing sugar. So formation of maltose. So I said this, it's 1,4. So carbon 1, position 1 of one glucose molecule and position 4 of another glucose molecule, participating through the process of condensation, leading to the formation of alpha 1,4 glycosidic bond. So these are alpha glucose. Remember, alpha position, alpha glucose. So alpha glucose, position 1 and position 4 forming the 1,4 glycosidic bond. The breakdown or hydrolysis of maltose will give us alpha glucose and alpha glucose, so 2 monomers. So this is simply the same illustration of the alpha orientation and the beta orientation of maltose. So when you look at this maltose, it's an important intermediate during the production of fermented beverages. So things like beer, during the fermentation of beer from cereals. So for example, when you're making beer from barley or millet or maize or rice, this maltose is a very important intermediate during that process. So basically, these are the disaccharides. So on the disaccharides, remember what monomers form a specific disaccharide. What monomers form a specific disaccharide? So you should know what monomer will form maltose. So of course, you know that, okay, this is going to be formed by alpha glucose and another alpha glucose molecule. And then you need to know what kind of glycosidic bond is formed. So you need to know the carbon positions that are participating in the formation of the glycosidic bond. Know the carbons that are participating in the formation of this glycosidic bond. Then know which ones are the reducing sugars, which ones are the reducing sugars. So you should know which monosaccharides are reducing sugars, which disaccharides are reducing sugars as well. So these you need to know. And then also, because you've known the monomers, you're also able to decipher if you are given a disaccharide. If you're asked if a disaccharide is hydrolyzed, what will be its product? What will be its product? So you're able to decipher since you already know the monomers that brings about the specific disaccharides. And also know that their common names. You know, lactose is milk sugar. Sucrose could be cane sugar. So just know those common names and also their functions. What do they do? What are their applications? What is their function? What is the function of sucrose? What is the function of maltose? So you need to know those. Are there any questions?
No, sir.